Okay, hello, good evening. Oops, Sebastian, haven't seen you. Hi, Joe. Should I call Joe, Sebastian? Yeah, I remember you. Wow, it's been a while. How are you doing? All right. Usually there are like 5 people or 4 people this class. I don't know where are they. Most of them, I think they returned to physical already. So I think it'll be less people, and turns out there's only you today. And I think some of them probably they are late from school. All right. So anyhow, let us continue with our revision this week. Oh yeah, by the way, May it's gonna be a revision month, okay, to prepare you for your exam, the first exam. So we will not enter anything new for this week. Supposedly we stopped until I think halfway through chapter 5. Okay, I don't know whose class you attended lah. I actually just started chapter 5 the week before we started our holiday. Right, but anyhow, we are going to chapter 1 today for our revision. Okay, so this is our chapter 1 form 5, which is the microorganism chapter. Okay, so this month, this year there will not be any seminar because usually there will be seminar for form 5. Okay, we will be doing revision in the seminar and we'll do normal class and as normal. But this I don't know why. Okay, maybe something wrong with the schedule. Oh, you just finished rate or reaction? Is it? Whose class are you from, by the way? Rate of reaction, that is chapter 4, was it? Was it chapter 4? A new teacher. Boo, is it? Okay. Chapter 4. I see. Because we just because the week right before we go for holiday, I actually started chapter 5. But anyhow, all right. So today we will be in chapter 1. I will try to go through this as fast as I can because you have learned this already, and I will try to speed this up because this is just a revision class. All right. So as you know what microorganisms are, all right, this one you already know. All right. You need to see them with a microscope. And what is a flora? All right. Flora, they are just normal, healthy microorganism you found in your gut. All right. The one in your stomach. So this is why you have heard of probiotics. When you eat yogurt, when you eat like a drink like Yakult, okay, you replenish that flora in your stomach, in the digestive tract. All right, so what are their functions? Okay, this is just a touch and go thing. Don't focus too much on it. All right, so it competes with pathogens to obtain nutrient and prevent the formation of colonies of pathogens. That means if you have a lot of bacteria, good bacteria in your gut, if you happen to eat something bad, all right, if you happen to eat a certain pathogen, that pathogen is most likely not going to survive because you have a gang of bacteria in your gut, so they are going to be bullied. That pathogen is going to be bullied and they can't survive and they can't cause you any stomach upset disease or whatever. So this is a good thing. All right, so it synthesizes vitamin B12 and K for you. Stimulates the growth of body tissue such as colon tissue so that you don't get colon cancer. So stimulates the production of antibody that fights pathogens. This is why if you have a normal flora, usually you won't get any funny stomach disease. All right, you don't get diarrhea for like no reason. This is why you don't simply eat antibiotics. So I think you know this already lah. If you simply eat antibiotics, you are going to bombard this flora. You will kill everything, which is not a good thing. All right, so do not eat antibiotics if you can help it. Okay, anyway, so this normal flora, okay, just read this for yourself lah. Don't need to memorize the species here. That is not your work. All right, and as for our microorganisms, we have 5 types. Okay, we have our fungi, which is the thing found in dark places like your mushroom, okay, like your yeast, and then algae, which is the plant found in fresh water. Protozoa, they are like tiny animals. Bacteria, they are, bacteria. And of course, virus is the only non-living stuff here. Virus is non-living. They are not a living organism. They are just some kind of protein that wreaks havoc in your body. that makes you sick. All right. So think of it this way. This algae is a small plant. All right. And this protozoa is a small animal. Very easy. So protozoa is like a tiny version of us, and algae is like a tiny version of a tree. Really simple to understand. All right, so you don't need to memorize the size and the shape. This is none of your concern. I mean, yeah, sometimes they'll ask for the shape of the bacteria, but hey, it's not worth your time. I mean, if you have time, you can go and read it yourself lah. The focus would be our nutrition and our methods of production. All right, habitat is fully depend on nutrition. So how do they feed themselves determines where do they live. All right, for example, if you look at there are three words for you to take note. There is a word called saprophyte, and then there's another word called parasite, and then there's another word called photosynthesis. So there are three words that you need to know. Photosynthesis means they make their own food. Saprophyte means they get something from dead organism. Parasite means they get something from living organism. All right, so these two, they are really close. Very simple to understand. You are still living now. If there's something growing on you, that's a parasite. But if you die, okay, if you happen to die and something to grow on your dead body, that is a saprophyte. That's the difference. All right, so fungi, they can either be saprophyte or parasite. So it depends on the type. But no fungi will do photosynthesis. This is why you don't find fungi in bright places. As long as there is sunlight or any strong light, you don't see fungi because they don't like sunlight. All right, so this is why they grow in dark and moist places. Whereas algae, it's the total opposite. They love doing photosynthesis, so they love to live under the sunlight. So you'll find them in fresh water or tree bark. Okay, and then of course your protozoa, this is a very funny animal because this animal, they can either do photosynthesis or do parasitic, or they can also do saprophyte. So protozoa can be all three. Because protozoa is a very broad range. This is the most complicated group to study because they come in many shapes and sizes. They come in many ways of feeding themselves. So they can live anywhere. Right? They can live anywhere. They can live in fresh water if they need the sun. They can live in fresh, moist soil if they don't need the sun. So it depends on how they feed themselves. All right. So the next one, bacteria, same as your protozoa. They can be all three. Photosynthesis, they can do photosynthesis. They can be parasites. They can be saprophyte as well. So this determines where they live lah. And virus, this one, no, they don't feed themselves. They don't eat. They don't do photosynthesis. They don't do saprophytism. They also don't do parasitism. No, virus can only reproduce in a host. If you are a host, they will come into your body and then they will use your body to reproduce. This is how COVID spread itself. So virus is a very funny thing. All right, they are technically speaking, they are not an organism. They are not a living thing. Okay, so anyway, methods of reproduction, this is very simple. For the middle tree, okay, virus, they don't reproduce, okay? Oh, besides if there is a host, the middle tree, they are all the same. They can either do asexual, that means they reproduce by themselves, they copy another one of them by doing binary fission. Binary fission, binary fission. They all do binary fission. This is the asexual. They can also do sexual, which is conjugation, conjugation, conjugation. So the middle tree, they reproduce using the same method. Virus is another thing, right? They don't reproduce. And for fungi is a very funny one because they do neither. Yes, they do conjugation. Yeah, they do conjugation, which is the same as the other three. But for asexual, for example, like yeast, yeast, they don't do binary fission. They will do something called budding. Which is this thing, they grow another tiny baby here, and then they break off lah. So that's your budding. All right, so for mucor, this thing is found on your bread. So if you don't eat your bread for like 2-3 weeks, they go bad lah. So this is how they reproduce by using spore formation. Pembentukan spora. So fungi is the funny one. All right, do pay attention on your yeast. All right, this is the most common microorganism they ask in exam. It's always yeast. You always see yeast in exam for some reason. So pay attention to that. All right, so anyway, this is not really difficult. I mean, it looks like there's a lot of things to memorize, but I would say like 50 to 70% of them, they are just useless information. You just need to know the nutrition and the methods of reproduction. You are good. All right, so let's keep our problem simple. All right, anyway, let's look at our factor. Arguably, I would say this is more important. I would rather you not memorize anything about the microorganism. I would rather you memorize the factors. This is for sure coming out. So I know it sounds funny. This is like a very small part in this note, but this is the most important part. So what affects growth of microorganism? What determines whether microorganisms grow well or not? There are 5 factors, all right? You have your humidity, how wet somewhere is, light, nutrient, temperature, and pH value. All right, so let's talk about humidity first, right? Humidity, I don't care what organism you are, as long as it is humid, they will grow well. All right, there is no microorganism that doesn't like water. So if there's water, humidity, they will grow up. Very simple. So light, this is very dividing. All right, so like I said, fungi, they hate the sun, whereas algae, they love the sun. So this is why light is a very divisive thing. Some they like it, some they don't. So if they do photosynthesis, yes, they like it. So bacteria and fungi, when they don't do photosynthesis, they live in the dark. All right, and of course, nutrient is the same as humidity. Always when nutrient increase, growth increase. That's all. So next, temperature and pH value. These two, they are the same. It's always about the middle. You see, humidity and nutrient is higher the better. But temperature and pH value, it's not. It's not higher temperature, higher pH value, the better. No, they need to be in the middle. Temperature needs to be 35 to 40. pH value needs to be neutral. They like the middle. So anywhere too low or anywhere too high, microorganisms, they don't grow well. So for example, temperature, you need to be this. Too low, they will retard the growth. Here's the thing, temperature, if it's too low, if it's too cold, let's say you put your food into the fridge, bacteria, they don't die, they just become inactive. This one you know lah. So if you freeze your meat, let's say you buy a piece of chicken, you freeze it, the microorganism, they don't die, it's just that they become inactive. So when you defrost them next time, they become active again. So low temperature doesn't kill, but high temperature, yes, they can kill. All right, so pH is almost the same thing. All right, so okay, pardon me, my caffeine is I had coffee this morning, so it's kind of crashing right now, so I'm yawning non-stop. Okay, so a pH value, if it is too high or too low, if it is too acidic or too alkaline, then they die. Very simple. Okay, so useful microorganism, this one, you can just also touch and go, don't need to focus too much. If you want to focus on yeast again. Yeast is always our main character because they're just too useful. What are they used for? They are used to rise bread dough, okay, or make cake, or to make alcoholic drink. This process is called fermentation. All right, this is how you make alcohol. This is how you make beer. You need yeast to make beer. All right, you just close them and let them ferment, then you will get your beer. So really easy here. So as for the next one, let's look at some of it. Lactobacillus is found in your yogurt. Bifidobacteria and lactobacillus, this is different lactobacillus. These two, they are used to remove skin such as fats and hair so that you can get leather. For example, if I'm LV, you know LV, right? Okay, they produce a lot of bags every year. They will use cow hide, okay, cow leather to make their bags. But you can't just use cow leather just like that without removing the fats and the hair. It will be disgusting. So this is how they do it. They will use this bacteria to peel off the skin and the hair so that the leather becomes okay to use. All right. So agriculture, for animal digestion, all right, if you're a cow, if you're a herbivore, you have all these good bacteria living inside your gut. Okay, these bacteria, they will produce cellulase. They will digest your cellulose into glucose. That's how it works. All right, cellulase is an enzyme, by the way. This is an enzyme to help you digest your plants. So this is why we can't digest veggie. Cows, they can. because they have the bacteria. We don't have it. So this nitrifying bacteria, you actually learn this in chapter 2. Don't worry, we will revise this next week. You just need to remember nitrogen fixing bacteria. Don't need to memorize the species here. What is nitrobacter and nitrosomonas? Not your thing. Just remember they are nitrogen fixing bacteria. What is nitrogen fixing again? Is to convert the nitrogen in your air, 78%, to convert them into the nitrogen in your soil so that they become fertilizer, so that plants can grow well. That's your nitrifying bacteria. And then, of course, you have hormones like insulin, all right, they are made from bacteria as well. And you have antibiotics like penicillin made from bacteria as well. Even your vaccines, they're made from virus. All right. Chapter 2, yes. Okay, that big circle and the small circle thing, right? There's a diagram of your nitrogen cycle. That one is one of the most important part in your chapter 2. All right. So this is your useful microorganism. Really just touch and go lah. Don't need to memorize so much here. All right, so let's move on. Let's go to our this eco thing, biotechnology and eco enzyme thing. I'll just read this yourself. I'm not going to talk about this. All right, not very common in exam. Okay, next, aseptic technique. This is how you get rid of microorganism. How do you remove microorganism? So sterilize it. You have 4 methods here, 5 in fact, sorry. Sterilization, boiling, antiseptic, disinfectant, and ionizing radiation. So the rest you can read it yourself. I want you to focus on these 2. Right? Usually in exam is these 2, antiseptic and disinfectant. So what is the difference? Very easy. Antiseptic, you can use it on human skin, whereas your disinfectant, you cannot use it on your skin. I mean, it's not very good for your skin lah. If you don't want to use it, okay, you can. But antiseptic will be the more correct choice, right? For example, acryflavin, which is the yellow medicine, you use it in your primary school. If you fell down, they will put some yellow medicine on your wound. That's acryflavin. The rest like Providon, isopropyl alcohol. Yes, your hand sanitizer can be used on your wound. That wouldn't be very pleasant, but hey, at least you can put it on your skin. All right. So whereas your skin, the disinfectant, you cannot use it on your skin. For example, bleach. I mean, it would be very unpleasant if you use bleach on your skin. So you can use this on surfaces like tables, floor, whatever surface you want to remove microorganism. You can use disinfectant. All right. So anyway, this antibiotic, like I said, they are not effective against viral infection. Antibiotic, they don't kill virus. But if you use it for no reason, you will develop something called antibiotic resistance. All right, it is when your antibiotic lose the ability to kill bacteria. Bacteria becomes resistant to it, which is not a good thing to have, right? This is why you don't take antibiotics for no reason. I mean, unless you have bacterial infection, then yeah, you need it. But other than that, no. All right, so treating infectious disease, pneumonia is a bacteria. You can use antibiotics. Athlete's foot, they are caused by fungus. You can use an antifungal cream. Shingles, they are caused by virus. You can use an antiviral drug. Right, that's all. Okay, really easy for chapter 1. Okay, so let's go into our exercise. I'll try to do as many exercises as I can lah. Sorry, this is chapter 1. Yeah, here. All right, so let's go to question 1, page 18. We will be revising a little bit of form 4 also for the next 2 weeks. 2 weeks later, next week we are going chapter 2. So anyway, question 1, classify microorganism P, Q, R, S. So P is definitely your yeast, so they are fungi. Q is paramecium, this is your protozoa. R is amoeba, protozoa also. S is your mucor found on your bread, so this is your fungi as well. So this Q and R, they are protozoa, they are tiny animals. You learned this in form 1 actually. They are found in fresh water. These 2, they are animals. They are our close cousin. It's just that we are much bigger in size. They are smaller in size. So the other 2 is your fungi. So state the microorganism used in bread making. Well, obviously is your yeast, which is P. Don't name the thing. They are not asking you to name. They just want you to state, so you give back the alphabet, do not say yeast, because if your name is wrong, then your answer here is wrong. Anyway, so B, diagram below shows a type of microorganism living in a bright aquatic environment. Whenever you see bright, you know they like the sun, and you know they do photosynthesis. In fact, this thing is called your Chlamydomonas. Yes, funny name. This is what you learned in Form 1. So what is the classification for this microorganism? Obviously, this fella is an algae because they love the sun so much. So what is the method of nutrition? So what do? They do photosynthesis. So explain. How do you know they do photosynthesis? Because they contain chlorophyll. They have chloroplast or chlorophyll. You can either use plus or fill. Either case is accepted. So next, number 2. So it's a type of microorganism. This is actually your yeast. Okay, I know it doesn't look like your yeast, but these are their buds. Okay, they are budding. So name the above. So if they ask you to name it, then you name. So yeast. Briefly describe the reproduction process. So they will do budding. Right? So they want you to describe. So they will grow new buds. These things are called buds. It's like a baby plant. Baby yeast is bud. See, how does this type of microorganism obtain nutrient? All right, they do saprophyte. All right, they're saprophyte. They're saprophyte because they feed on dead stuff. Yeast don't live on living organism. They will feed on dead stuff like sugar. Sugar is dead. They feed on flour. Flour is dead. This is how they raise your bread. They don't eat living stuff. They eat dead stuff. So if they eat dead stuff, it's called saprophyte. If they eat living stuff, then it's called parasite. That's the difference. Okay, so far it's still so easy. So far it's still very easy. So number 3, diagram below shows an experiment to carry out to study the factors affecting the growth of bacteria. So let's look at A. This one is moist and nutrient. Moist and nutrient agar. So I believe A will grow very well because it has moisture and nutrient. Whereas B, there's only dry and nutrient. There's no water. Type of microorganism, isn't it fungi? Oh yeah, wait. Wow, sharp eye, you have that. Yeah, I think it's fungi. I thought it's named the microorganism. Oops. Sorry. Okay, so dry as in no water and this one is moist without nutrient. Okay, so mark for the factors present for the following petri dishes. So A definitely has both nutrient and humidity, whereas B there's only nutrient. C there's only humidity. There's no nutrient. So obviously B and C the bacteria, they don't grow very well, but in A they will grow very well because it has both. So in your opinion, which petri dish has the fastest growth of bacteria? Explain your answer. So definitely A, because it contains nutrient and humidity. Okay, or water. All right, like I said, nutrient and humidity is in the case of the more the better. But temperature and pH is not. They need to be in the middle. So state one other factor affecting bacterial growth. So there's 2 nutrient and humidity, 2 factors here. You have another 3. So anything you want, pH, temperature, or light. You choose one. I'm giving you 3 here because there's 5. So yeah, light. Not just sunlight, it can be any light. Like our artificial light works as well, not just sunlight. So number 4, diagram below shows an experiment to study the effect of pH on growth of bacteria. So P is acidic, they put acid here, and Q is alkaline, R is distilled water. So from your knowledge, you also know R is going to grow the best because this thing is neutral. Neutral. So after 2 days, by looking at the observation, you know So if it is acidic or alkaline, they are slightly cloudy. Cloudy means how dirty they look. The cloudier they are, the dirtier they look. That means the better bacteria grow, because bacteria is going to make the water look dirty. So as you can see, R is very cloudy, very dirty. There's a lot of bacterial growth there. So state the variables. MV, what is that you're trying to control? You're trying to control pH value. Right? Constant variable. So you're keeping one thing. You're changing one thing. The other four you don't change, which is your light, okay, humidity, your temperature, and your nutrient. So I will stick to temperature because temperature is really easy to write. So all three you need to keep them in temperature in 37, because you see humidity it's a bit hard to accept because all three of them they have water. So you can't really write humidity here, but temperature is definitely correct. This is constant, things that don't change. Whereas this is your RV. Cloudiness of the mixture is your RV, your responding variable. So B1, state the optimum pH value for the growth of bacteria, definitely pH 7. So what inference can be made from the result? So what is inference? Inference means explain your observation. All right, explain your observation, explain what you see. So what do you see here? You see R being the very cloudy one. All right, so PQ is the non-cloudy one. So what explains this? You can say bacteria grow the fastest in neutral condition. Neutral condition. So neutral, they grow the best. Acidic or alkaline, they don't grow really well. This is how you explain what you see, because PQ, acidic and alkaline, they are only little cloudy, whereas your R is very cloudy. That means they grow very well. All right, so C, from the result, state the operational definition. Again, you have this thing. So I don't know whether you still remember how to write this. Operational definition means they want you to write the definition based on your RV, based on what you see or your observation. You can either base it on your RV or base it on your observation. They are the same. So if they're asking for the definition of something, all right, for example, they want you to state the definition for growth of bacteria. So what's the first step? You write back growth of bacteria. Since they want the definition of growth of bacteria, right? So you say growth of bacteria is, this is the first step you write. And then second thing, you explain it to me. Is this a process? Is this an object? Is this a thing? So growth is a process, right? So growth of bacteria is a process. And base this on your RV. What is the RV says? The RV says the cloudiness of the mixture. So if there's a lot of growth, mixture is very cloudy. If there's very little growth, there's very little cloudiness. So growth is depending on the cloudiness. So the growth of bacteria is a process that causes mixture to be cloudy. So you write this based on your RV, based on your observation. All right, so 3 steps to write your operational definition. Let me repeat this again. Number 1, you write back what they ask. Number 2, you state whether is this a thing, an object, a process, a factor, and whatever. And then number 3, you write based on your RV or your observation, which in this case is the cloudiness. It causes the mixture to be cloudy. So that's your operational definition. Not very hard to answer And even if you can't answer a question like this, hey, it's only 1 mark. All right, don't sweat. It's okay. In your exam, maybe they'll ask for 2 operational definition for 2 questions. So there's only 2 marks there. I mean, it's really easy to score, but if you don't score, it's still fine because it doesn't carry a lot of marks. So next, number 5, diagram below shows the role of useful microorganism in life. So this is your nitrogen fixing bacteria. This is the bacteria that produce cellulase for the cow. This is to make insulin. So name the bacteria involved in P. So this is your nitrogen fixing bacteria. There's no need to go crazy and say, oh, Nitrosomonas sp and Nitrobacter sp. If you are taking biology, then yes, but your science, okay, it's okay, doesn't matter. So number two, state the function of this bacteria in agricultural field. Like I said, it converts nitrogen in the air, okay, into nitrate in the soil. So nitrate bacteria pengikatan nitrogen. Oh, you're writing it BM, is it? Okay, pengikatan nitrogen. I can write BM for you if you want. Okay, bacteria pengikatan nitrogen. Menukarkan nitrogen di dalam udara kepada nitrat di dalam tanah. So they will convert that 78% into your soil. So as you can see, this is very important, because if they don't do this, your plants cannot grow. If your plants cannot grow, then that will be the end of life. That's how important they are. All right, so name the class of microorganism involved in R. So R, who makes your insulin? Insulin is made from bacteria. All right, insulin is the thing you used to inject into people with diabetes, whether type 1 or type 2. If some people, they are born without the ability to make insulin, then this is how they make it. All right, so funny thing about this insulin, insulin is very cheap. All right, insulin is very cheap, and it's like a potential lifesaver. Last time, not too long ago, which is like 100 years ago, if you are born with type 1 diabetes, that means you cannot produce insulin, you will die at a very young age, because sugar kills you. But nowadays, if you are born even with a type 1 diabetes, you cannot produce insulin. Hey, it's not the end of the world. They will give you insulin identical to what everyone produced, same as what everyone produced, because they use the bacteria to produce this. All right, insulin is a basic medicine that everyone should have access to. All right, and which is a joke, because in the US, insulin costs a lot of money. All right, insulin, I think, okay, here's the thing, in the US, it's a very money-minded society. All right, insulin over there can easily cost you up to $400-500 a week. All right, so you would say, how can someone make money from selling? It's like selling Panadol like at 10 times the price. This is what they do. Okay, very scummy practice of their society. So this is why in the US, you better not get sick, or else, unless you're rich, that's a different story, because no, their insurance doesn't cover this thing. So if you have diabetes and if you're poor, well, be prepared to die. All right, they call themselves the best country in the world. My food, they cannot even afford, they cannot even let their people afford insulin. All right, so be thankful. Malaysia, at least our insulin is cheap. So C, how does microorganism help the digestion of food in Q? So they will produce cellulase. Okay, this thing will digest cellulose. All right, so cellulose is your vegetable, your grass. So do not say that, sir, how come cows, they eat grass, they get fat? I eat grass, I don't get fat, because you have no cellulase. Okay, so that they can digest grass and absorb their nutrient. We can't. It just passed through us as fiber. So anyway, D, diagram below shows the type of microorganism used in processing industry. Again, you have your yeast. So name the process. This is your fermentation. All right. Yeast is a multi-billion-dollar industry. That's how they make beer, make alcohol. So state the gas released during the process name in D1. So fermentation will produce carbon dioxide. The gas in your beer, it's that carbon dioxide from the yeast. So this is how they rise the bread dough. Right, when you put yeast on the dough, they will produce carbon dioxide, which makes the bread fluffy and nice to eat. That's the carbon dioxide lah. All right, so anyway, number 6, diagram below shows the result of an experiment to study the effect of different concentration of antibiotic on the growth of bacteria. So as you can see, this is a petri dish, right? This is a petri dish, and you see like dots on it. These are the bacteria. And when I take two discs, I soak it in antibiotic. One, I soak it in high concentration. Another one, I soak it in low concentration, and I put it there. What is this? What does this clear area mean? This clear area means there's no bacteria. So as you can see, if I use high concentration, there's bigger clear area because I kill more bacteria. If I use higher antibiotic, more bacteria will die. That's why there's a bigger clear area. Whereas if I use a lower concentration antibiotic, I'm going to kill less bacteria, so there's a smaller clear area. Very easy concept to understand. So write down one observation for the above experiment. So you can say the clear area for antibiotic this of, wow, this is so long to write, okay, of high concentration is bigger than low concentration. I'm just keeping it short. I don't want to write the whole thing. Wow, this is such a long word to write. So basically, this big area, this clear area is bigger than the big area here. All right, so write one inference. So explain why is this bigger and why is this smaller? Okay, because higher concentration of antibiotics, kills more bacteria. Okay, so you have a larger clear area. Okay, the higher the concentration, the more the bacteria dies. That answer, I would save it for hypothesis. Don't try not to use the higher lower one, the higher the more one. That is for hypothesis, because if you write this answer, if they ask for hypothesis, you have to repeat your answer. So try not to. So again, higher concentration means they kill more bacteria. They have a larger clear area. So basically you explain what you see. No need to compare. So what is the responding variable? So what's your RV? RV is the area of clear area. Okay, now, I know it sounds funny. Luas permukaan kawasan jernih. BM makes more sense. All right, so based on this experiment, state the operational definition for antibiotic. So again, like I told you, operational definition for something, you write the something first. So antibiotic is what is it? Is it an object? Is it something? So antibiotic is a substance. All right, it is a substance or a chemical substance. So what do you see? You see when there's antibiotic, there's clear area, right? So antibiotic is a substance that creates a clear area, right, in a petri dish of bacteria, in a petri dish of bacteria. So if you want to write in a petri dish, you just create a clear area, that's all. So antibiotic, the first step, you write whatever they want you to define. Okay, they want antibiotic, so you write back antibiotic, and they'll tell me whether is this a substance or an object or a process or whatever. And then the next one, you write this definition based on your RV or based on your observation. Really, really simple. Okay, so again, they're asking for operational definition. You'll be seeing this a lot, So fret not. After you do it for a few times, it's not as difficult as you think. So mark for the microorganism that can be destroyed by antibiotics. Definitely your algae. Definitely, this is your bacteria, by the way. This is algae. This is virus, so definitely not a virus. All right, so next, number 7. Diagram below shows the clinical procedure performed on a patient who needs to be given an injection. So that's the shoulder. Name the material applied to the patient's shoulder. This is alcohol. Or just write it nicer, isopropyl alcohol. Isopropyl alcohol. This is an antiseptic, right? Not disinfectant. Right? Disinfectant is not for skin. So in your opinion, what is the purpose of applying the material to the patient's shoulder first before injection is given? This is to sanitize. If you don't want to say sanitize, okay, write sterilize. Sterilize would be more suitable. Sterilize the area, right? Or you can say clean the area from microorganisms. Because I mean, if you don't do this, you can still inject, but if there are bacteria or microorganism on the skin, and if you puncture the skin while you inject, there's a chance of infection. I mean, the chance is low, but it doesn't mean it's 0. So to not take our chances, that's why we better do this first. It's just good practice, right? So B, what do you think will happen if the above procedure is not performed before a person is given an injection? All right, so you can say microorganism might enter through the wound. So might cause infection. So when you have an infection, how do you know you have an infection at your injection site? Very easy. After your injection, you'll get a pus. You'll get like a big pimple there, like a boil, like a swollen thing there, and if it's full of liquid, if there's a pus there, you definitely know there's an infection. So usually it doesn't happen Usually it doesn't happen After your COVID injection, you just kind of feel a little bit of soreness. That's all. But if there's a big injection, if there's a big boil, if something is growing on the skin, and the thing is full of pus, it looks like a pimple, that definitely is infection. All right. So I'm sure you can tell what an infection is. Your skin will not look good. So give one other situation where the above material and procedure is suitable to be used. So where can you do this? One other situation. Where else can you use this? I couldn't think of any though. Besides injection, the material and procedure. So where else can you use isopropyl alcohol? Thermometer, I guess. Okay, so you can say sterilizing. Okay, sterilizing your rectal thermometer before use. This is in your form 4. Right, so you learn there's a thermometer called rectal thermometer where they shove the thermometer into your butthole. Yes, you can use that. You can use this here. You can clean the tip using alcohol and insert it to said butthole. So same thing. This is to sterilize, but I don't think you use. I mean, rectal thermometer we still use it, but it's mostly for babies lah. We don't use it on adults unless it's a personal preference thing. Hey, no judgment. So D, take the examples of material that have the same function as above. So won't burn. Good question. I don't know. Don't plan to find out. Okay, so your acryflavine and your povidone. All right, so chlorine and hydrogen. Okay, do you know what is hydrogen peroxide? This thing is used to dye your hair. This is a very strong bleach. Also another type of bleach. So do not let this thing touch your skin or let this thing touch your shirt. This thing can bleach your shirt. If you happen to drop a drop of hydrogen peroxide on your t-shirt, this thing will strip away the color and then you will get a spot there. That's the hydrogen peroxide. Hey, some crazy fellas, they use this hydrogen peroxide to whiten their teeth. I mean, do whatever that makes you happy lah. They will just apply on their teeth and then they will just wash it off after a few minutes. And they say it's supposed to whiten your teeth. I mean, come on, your teeth is not supposed to be white. Your teeth is supposed to be slightly yellow. Do not be cheated by all these social media and Hollywood and K-pop or whatever. Your teeth is not supposed to be white. All right. If you look at those celebrities that have white teeth, it's because they have something called ceramic implant on their teeth. Basically, they go to the dentist, they put a piece of ceramic in front of their teeth so that white is from the ceramic, not from their teeth. I mean, if you have the money, sure, why not? Go ahead. And if you're a celebrity, you use your face to cari makan, okay lah. But if you're not a celebrity, what's the point of doing it? Right? Your teeth is supposed to be slightly yellow. I mean, not as in the smoker yellow lah, but you get my point. So number 8. So this is caused by a virus. Oh, this is called shingles, by the way. Shingles is a very nasty skin infection. It's caused by a virus. Very rare, but it's sometimes it will happen. So this is called shingles. All right. So what method can be used to treat the disease? Okay, you can apply your antiviral cream. Do not use your antifungal cream. Oh, sorry. Yeah, here. So this clotrimazole. Oh, I use this all the time. Sometimes I get infection by fungus. I just use this lately on my skin. Okay, I went to the gym and so I didn't wipe the equipment and somehow I got some nasty infection. It was just a small spot lah. I use this. All right, but you do not use clotrimazole on your shingles. It doesn't work because this is supposed to kill fungus, and this S-cyclovir is supposed to kill virus. So they work in different ways. So doesn't work there. So explain why antibiotics cannot be used to treat the disease, because antibiotics does not kill viral or virus or viral infection. All right, so D, patients who take antibiotics are always told to finish the antibiotics rather than stop taking the antibiotics when they feel better. Explain why. This is to ensure all microorganisms are killed. This is to be safe, to make sure they are killed. Okay, so that microorganism does not reproduce again. So if they reproduce again, they will cause a second wave of infection. So that's what happens when you don't finish your antibiotic. You should always finish them. Okay, it's a course. You finish one course, and if it doesn't work, you go for another course. You don't like stop halfway. So E, Mr. Jamal is suffering from tuberculosis. This is a bacterial infection. Tuberculosis is caused by bacteria. It infects your lungs. This thing is much higher death rate than COVID. COVID's death rate is only like 0 something percent, 0 point something percent. Tuberculosis is 10. That's like 100 times stronger. So he is being quarantined and given antibiotics for treatment. Mr. Wong is suffering from dengue. He's not quarantined and not given antibiotics. Explain why the treatment for Mr. Jamal and Mr. Wong is different. All right, so why do you need to quarantine this? Okay, you can say tuberculosis can be spread via air. All right, so we can spread via air. Therefore, quarantine is useful. Okay, so given antibiotic, because you can say antibiotics kill bacteria that cause tuberculosis. This is why they quarantine. This is why they use antibiotic. Whereas for Mr. Wong, okay, dengue is spread by mosquito bites. So quarantine quarantine is not effective. There's only the quarantine. You kill the vector in this case, you kill the bacteria. So, sorry, you kill the mosquito. So, antibiotics. Wow, this is a long question. Antibiotic does not kill dengue fever. So 1 point for one explanation. This is why they give you 4. You usually don't see this kind of long question in exam because it's too much work to answer. So usually it will be only like 2 to 3 points. 4 is too much. Okay, so antifungal is used to treat fungal infection. All right, if you need me to slow down, let me know. I'm just quickly bringing you through because it's very straightforward at this point. So which of the following is an example caused by fungal infection? Okay, so definitely your athlete's foot. What is athlete's foot? I don't know how you call this. It's called ringworm actually. The scientific term is ringworm. That means your foot is you have some nasty spots on your foot, usually caused by fungus. So 2 factors that may cause fungal infection. You might say direct contact. Okay, direct contact with direct skin contact with infected individuals. So if someone is infected, you touch their skin, you get it lah. So direct contact. 2 factors that may cause fungal infection. Direct skin contact, and of course, if you can say, you want to say open wound. Open wound, it contributes as well. So state one difference between antifungal and antiviral. This thing kills fungus lah, whereas this thing kills virus. Really easy. Why is this even a question? So anyway, let's move on. Okay, you know what? This thing, if you want, I don't think I'm going to discuss. This is too long already. I mean, if you want the answer, I can put it on Google Classroom. You can check it from there next time. You can try and do this, right? This is the 10-mark question you usually see in question 10. That 10-mark question, the experiment question. So if you have the time, you can try this out. All right, so next week, we will go into chapter 2 revision, which is that nitrogen cycle thing. And then next week, we will revise on form 4. I think it will be chapter 11 and 12, if I'm not mistaken. All right, so I think I'm going to stop here today. Right, I hope you learned something new. So thanks for coming, although it's a very lonely class. I hope you don't feel that way. All right, so I'll see you next week. So take care. See you. Happy Wednesday. Bye-bye.